In essence, today, what I'm going to be doing is quantifying the maximal and minimal rates of denitrification in a wetland along the Mississippi River. Maybe. But first, I'd like to frame for you a larger problem of eutrophication, particularly within the central United States, in which this eutrophic nutrient load can reach as high as 3 milligrams per liter in terms of total nitrogen, and then 0.3 milligrams per liter in total phosphorus. Now, this eutrophic nutrient load is particularly problematic as it gets to the southern United States and the Gulf of Mexico, at which point it begins to receive warmer temperatures and ultimately contributes to the blooming of potentially toxic algal populations, in which these populations die off and ultimately decompose, reducing the dissolved oxygen to less than 1 milligram per liter, ultimately contributing to the dead zone in the Gulf of Mexico, which currently stands at about the size of the state of Connecticut, so quite substantial. And then, as a byproduct of this, we see the disruption of many local food webs, in turn causing disruption to the economic and ecological environment, ultimately reducing many of these to nothing. This isn't a problem that's just isolated to the southern United States or the United States as a whole. This is a global issue and is ultimately something that we need to grapple with on a global scale, both due to warming temperatures and increased anthropogenic development. As you can see by all of these red dots, we're seeing an increase in these algal blooms. But let's focus back on the Mississippi River Basin. Largely, this eutrophication is the byproduct of levee disconnection. We've disconnected more than 90% of the Mississippi River's floodplains, in doing so, disconnecting it from the river. This has in turn disrupted local hydrology and nutrient cycling, largely the byproduct of degrading, fragmenting, or ultimately removing many of the local wetlands for anthropogenic development. Be it for agricultural land or residential development, we're cutting off many of these existing wetlands. And these wetlands are significant because they have substantial nutrient retention potential. In the upper Mississippi River alone, we could retain nearly 12% of the nitrogen load currently passing through that region. Ultimately, though, this isn't as simple as surface water inundating these wetlands or these floodplains. We need to think about the groundwater movements as well, particularly under seepage and upwelling, as they can occur both with and without levees, but these are ultimately minor compared to the effects of surface flooding. Ultimately, though, because they occur both with and without levees, they can resaturate a greater portion of the floodplain throughout the year, as they are not restricted to just these high water levels. And they allow many of these wetlands to persist behind levees when not actively inundated with surface water. Next, I'd like to frame for you the microbially mediated nitrogen cycle as we'll look at it today. And this is a simplified version, but ultimately the focus of my research is on denitrification, a process that consumes nitrate and ultimately produces inert N2 gas. Now, this is a form of permanent removal from the aquatic environment, as this N2 gas is largely inert. We also have another form of competing nitrate removal in the form of dissimilatory nitrate reduction to ammonia, or DNRA. This is a process that consumes nitrate, but ultimately produces ammonia, which is still reactive and does contribute to eutrophication. And ultimately, this ammonia can then be converted back into nitrate through nitrification. But what kind of environmental conditions optimize it? Well, for these forms of nitrate removal, denitrification and DNRA, they're both optimized by very similar conditions. Those being low dissolved oxygen, alkaline pHs, an abundance of nitrate and labile carbon, but also warm temperatures, as many of these microbial communities are ectothermic. Next, I'd like to introduce our field site. This is the Dogtooth Bend, a peninsula along the middle Mississippi River, just northwest of the river's convergence with the Ohio River. Levee breaches since 2011 have reconnected parts of the floodplain to the larger hydrology of the river, and thus subject it to stochastic flooding. We selected wetlands to represent different sediment depositional environments across the Dogtooth Bend, but ultimately the question becomes, how do we replicate these dynamic environments in the laboratory? And here's a photo of the Dogtooth Bend following a major flood in 2019, in which these two large river barges were deposited in the agricultural fields. And as you can tell by the houses or comparison, these can be quite substantial. But ultimately, how do we measure this? Well, these are typically done in laboratory incubations, in which we use some kind of incubation water and pass it through a sediment core while measuring both the inflows and the outflows, thus allowing us to compare the flux between the two and evaluate what processes may be occurring. We further add a layer of control to this by doing it in an environmental chamber, allowing us to control temperature or light. But we added the novel approach, this experimental deep injection core, which you can see on the left-hand side. This sediment core uses a deep injection straw in order to insert water to the bottom of the sediment core, allowing it to pass through the entire volume of sediment before exiting the core. And this is designed to replicate natural processes like infiltration, exfiltration, or lateral flow of water through the sediment. And ultimately is designed to replicate many of these groundwater effects, which are largely neglected in traditional incubations. As you can see by the traditional incubation core on the right-hand side, the water is simply injected onto the surface of the core and allowed to diffuse downward into the sediment. Now, this fully replicates processes like surface water inundating a fully saturated floodplain, but it doesn't replicate much else than that. To test how these two incubation strategies may truly differ, we did a 96-hour rhodamine dye injection in which we compared how the dye permeates these two different sediment cores, one with the steep injection straw and the other using the typical approach. And as you can tell by the photo on the right-hand side, we see significant differences in how the dye permeates the soil. With the deep injection straw, the dye fully permeates the soil, whereas without, it only permeates about the first inch of the sediment. This ultimately led us to our research questions. First, how will water delivery, deep injection versus surface injection, affect our denitrification rates? How will these rates change under stagnation, be it oxic or anoxic conditions? Does sediment type change this effect? And then how does microbial denitrification balance with nitrate loss stoichiometrically? We also wanted to understand the environmental conditions that may promote denitrification, but also cause variation within those rates. And to do that, we used intact sediment cores collected from the Dogtooth Bend and incubated them with river water collected at Thebes, a site along the Mississippi River just northwest of the Dogtooth Bend. We incubated these cores at 30 degrees Celsius and did this for 4 days in the dark in a 2x2 factorial design in which we used the two sediment cores I talked about previously, the experimental deep injection core and the traditional core, but also both oxic and anoxic water as to fully replicate groundwater and surface water respectively. We then sampled N2 gas and geochemistries such as nitrate, ammonia, phosphate, pH, and alkalinity every 24 hours as to catalog the change that occurred within these sediment cores. And then sampled the N2 gas directly using a membrane inlet mass spectrometer, or a MIMS, which is shown here in the bottom right. And then following these incubations, we looked at the physical characteristics of the sediment, looking at things like ash-free dry mass, sand content, porosity, permeability, bulk density, and particle density. And then did many of our statistical inferences by multivariate, multilinear regression. Let's start addressing some of these research questions. First, how will water delivery, deep injection versus surface injection, affect denitrification rates? And then how will these rates change under those oxic versus anoxic conditions? And to do that, first I want to familiarize you with the graphs that we'll be looking at. Here on the y-axis, this is our measure of denitrification in milligrams per meter square per day of nitrogen. On the x-axis, these are our different treatment combinations. Starting at the far left in dark blue, this is our anoxic deep treatment, which would be our experimental deep injection core receiving anoxic water. Next to it in the light blue, our anoxic surface treatment, that would be the traditional sediment core receiving anoxic water. Then oxic deep, this is again, experimental deep injection core receiving oxic water. And then finally, oxic surface, this being the traditional sediment core receiving oxic water. But on average, we see the formation of this clear hierarchy in which we have our greatest rates of denitrification in our anoxic deep treatment and our lowest rates of denitrification in our oxic surface treatment. But ultimately, how do these rates change under oxic versus anoxic conditions? And as you've probably already seen, we see significantly higher rates of denitrification in these anoxic conditions. Now, this wasn't surprising, as I previously said, denitrification is optimized under these anaerobic conditions, but It's also inhibited under oxic conditions. But what about injection type? Deep injection versus surface injection. Well, as you can see in these red boxes, within each water type, our deep injection facilitates greater denitrification by supplying nutrients to the entire sediment core, as opposed to just the surface, which is shown here in the black boxes. All the while, we need to consider these oxygenation effects. So denitrification is always more significant in our deep injections. But how does this change with sediment type? Specifically, at a clay-rich site, we found that the clay ultimately limits infiltration, and our rates no longer differ with injection type. We do, however, see strong differences based on the oxygenation of the sample, the oxic versus anoxic. And this signals that the microbial communities here are very biologically active on the surfaces of the sediment, but the water is not infiltrating the entire volume. And you might be asking yourself, well, you're still injecting it deeply. Why isn't it interacting with the entire volume? Well, at these clay-rich sites, we see significant channelization, which allows this water to rapidly pass back to the surface, still not interacting with the entire volume of the sediment. And you'll note some environmental factors here on top of the slide. Those highlighted in green could significantly account for 78% of the variation in denitrification rates across our different field sites, within this field site. And these are ultimately the byproduct of our multivariate, multilinear regressions. And I'll return and talk about these later. Next, at our silt-rich site, we see that the silt-rich site allows for the most permeation of water through the sediment, and we now no longer see a distinction between our oxic and anoxic treatments within injection types. And this signals that there's a very robust microbial community within these sediment cores, and that it is able to consume the excess amount of oxygen within these oxic cores, but still be a very significant denitrifier, comparable to the anoxic treatment. We do, however, see a strong distinction based on injection type, with the deep injection facilitating greater denitrification by supplying nutrients to the entire sediment core, as opposed to just the surface in our traditional incubations. And these environmental factors could explain 71% of that variation. Ultimately, though, at a sand-rich site, we see the greatest variability in our denitrification rates, as the sand allows for relatively unrestricted flow of the water through the sediment. It's ultimately too coarse to support that same robust microbial community that consume the excess amount of oxygen at a silt-rich site. Thus, a sandy site must rely on outside factors in establishing those optimal anaerobic conditions in order to facilitate denitrification. But when those conditions are met, we see our greatest possible rates of denitrification. When they are not, however, we see our lowest possible rates of denitrification. And these environmental factors could explain 67% of the variation in denitrification. But let's just start addressing our fourth research question. How does microbial denitrification balance with nitrate loss stoichiometrically? And ultimately, as I evaluated this data, I was surprised with the results, as it indicated multiple metabolic processes occurring simultaneously and were far more complex than I initially had assumed. Going into this, I had assumed that this was going to be a fairly straightforward relationship in which 2 nitrate would be introduced, and ultimately 1 dinitrogen gas was going to be output. In essence, a 1-to-1 relationship. What we saw in practice, though, was not this 1-to-1 relationship, which I had assumed would occur. In fact, we see much more denitrification taking place than we do in observing nitrate loss. And here on the y-axis, this will be our measure of denitrification by field site again, whereas on the x-axis, this is our measure of nitrate loss in milligrams per meter squared per day. And if there truly was a 1-to-1 relationship, we'd expect these points, representing our different treatment combinations, to lie somewhere along these red lines. In fact, regardless of the treatment and the field site, we see significantly more denitrification taking place. So what factors could contribute to that, and how substantial was this difference in nitrate loss to observe denitrification? Well, to look at this, we took our denitrification rates and subtracted our observed nitrate loss to figure out exactly how much nitrate we were missing within our incubations. And we found that we were missing the most amount of nitrate in our anoxic cores, which were our most successful denitrifiers, and the least amount of nitrate was missing in our oxic cores, our less significant denitrifiers. Ultimately, though, we concluded that nitrification was the most likely culprit, in that it consumed ammonia and ultimately produces nitrate, and it could occur simultaneous to denitrification, preventing us from observing where this nitrogen is pooling. And it's important to note that nitrification is ultimately optimized by very similar conditions, those being warm temperatures, alkaline pHs, and moderate dissolved oxygen. It's important to note, however, that it is not inhibited by low dissolved oxygen as long as it was greater than 0.5 milligrams per liter, which our anoxic incubations were. Now, while this is the most likely culprit, it's important to note that this could simply be the byproduct of sediment binding and it just gradually being released over the course of this incubation. But since we see this effect across all of our field sites, regardless of sediment type, this seemed less likely than simple nitrification occurring. Next, I'd like to talk about our outliers. This is never as simple as we'd like it to be. This is our Grand Lake field site, another clay-rich site that's predominantly fed by groundwater, but not actively inundated with surface water. Here, we expected a similar trend to our other clay-rich sites, but ultimately found something fundamentally different. Our lowest average denitrification rates in a hierarchy that doesn't follow the trend of any of our other field sites, but this site still consumes a similar amount of nitrate as our other field sites. So ultimately, what could be occurring here? And we could still account for 44% of the variation in denitrification using these environmental factors. But ultimately, this led us to look at some other potential pools of nitrogen within our cores, specifically also at different metabolic processes, specifically dNRA. And again, that's the process by which nitrate is consumed and ammonia is ultimately produced. And here on the graph at the left, on the y-axis, we have our measure of ammonia production, again in milligrams per meter squared per day, and nitrate loss on the x-axis, again, milligrams per meter squared per day. But what we found when averaging our other 3 field sites together is that we have very low ammonia production, around 100 milligrams per meter squared per day. Regardless of treatment or field site, this was significantly lower. As compared to Grand Lake, our outlier, we have significantly higher rates of denitrification, specifically in our deep injection cores, with our highest observed rates of ammonia accumulation being in our oxy-deep core. And this made us think about some other processes that might facilitate denitrification or ammonia production, outside of dNRA. And we likely landed on decomposition as the main culprit here. In our anoxic deep treatment, though, we see much higher nitrate loss, and we still see significant ammonia production, indicating that this could still be dNRA. Ultimately, though, this is likely the byproduct of many metabolic processes occurring simultaneously, specifically dNRA and decomposition, as I've talked about previously, but also simple metabolic waste as the byproduct of cellular respiration, all resulting in increased concentrations of ammonia. And with this in mind, it changes our nitrogen cycle just a little bit. Ultimately, though, we would need some form of isotopic analysis to differentiate what processes were dominating in regards to this ammonia production. Next, I'd like to refocus on denitrification as it's the main subject of my thesis. Specifically, what environmental factors promote denitrification, but ultimately also how they cause variation in denitrification. Starting with sediment traits, as these seem to have one of the stronger effects. And sediment traits are going to encapsulate many different parts or many different aspects. To begin with, we'll cover particle size, which assists in establishing this ideal microbial environment for denitrification. Specifically, our largest particle sizes were sand, and these large particles allow for large pore spaces, which in turn allowed for relatively unrestricted flow of water through the sediment. This, however, leaves the microbial community exposed and ultimately lowers exoenzymatic activity as these particles are largely uncharged. What this means is that an outside factor needs to establish those optimal anaerobic conditions in order to still facilitate significant denitrification. A silt-rich site, by contrast, seems to be the ideal particle size, in that it still allows for the movement of pore water through the sediment, but it ultimately protects the microbial community while still allowing room for growth. And then finally, a clay-rich site, which is our smallest particle size. Here, we found that the clay ultimately constrains the movement of pore water, limiting the transport of fresh nutrients into the sediment, but it also constrains microbial growth, by simply being too small and tightly packed for these microbial communities to grow as effectively. It's important to note, however, due to its charged nature, it does facilitate greater exoenzymatic activity than any of our other sediment types. And we found that sand was ultimately a significant factor causing variation at all of our field sites, with coarse sand being significant at 3 of them and fine sand being significant at 2 of them. Next, in terms of sediment traits, we covered bulk density and particle density. Bulk density was significant at one of our field sites, and it's a measure of sediment compaction, as this could constrain the flow of pore water through the sediment, but also squeeze out some of these microbial communities. It's important to note, however, that this sediment compaction may also assist in maintaining those optimal anaerobic conditions, and that this is inherently closely related to those sediment particle sizes. As you can imagine, a clay would be most closely related to a very compact sediment, which would be represented here on the left side of this diagram, whereas sand would be a more coarse sediment, allowing relatively unrestricted flow of water through these pore spaces. It would be represented on the right-hand side. Ultimately, though, we also evaluated particle density. And this particle density was significant at 2 of our field sites and was used to differentiate different kinds of minerals, but also humic material within our sediment cores. And it assisted in differentiating both sand, silt, and clay in our cores based on the density of the sample. Ultimately, though, we found that all of our cores existed on some kind of gradient consisting of both sand, clay, and silt. It was just a question of whether or not one sediment type was dominant compared to the others. And this was significant at 2 of our field sites. Next, I'd like to evaluate dissolved oxygen, which was significant at 3 of our field sites and ultimately accounted for the greatest variation within our denitrification rates, specifically accounting for 20% of the variation we observed across all of our field sites and treatments. And it's important to understand the effects that dissolved oxygen could have, as it can limit denitrification in 2 ways, specifically by limiting denitrification by providing a more favorable terminal electron acceptor to nitrate, as it is a better terminal electron acceptor, but it can also inhibit denitrification directly by inhibiting the enzyme nitrate reductase. Dissolved oxygen, though, is inherently closely related to these sediment traits, as the larger the pore space, the more readily this dissolved oxygen can infiltrate the sediment and ultimately disrupt denitrification as it's taking place. Whereas if it takes place towards the bottom of this diagram, in those more compact sediments or finer granules, then it's more difficult for that dissolved oxygen to infiltrate the sediment and ultimately disrupt denitrification. Next, I would like to talk about the forms of nitrogen that we measured. First, we evaluated nitrate. Again, that's that terminal electron acceptor that we thought would be directly related to nitrate loss. And we found that it was ultimately never significant, causing no variation in our denitrification rates. Now, this was likely because it was abundant in our source water, or was ultimately replaced very readily by nitrification, or being released fairly readily by the sediment. Ammonia, though, was significant at 1 of our field sites and could have been the byproduct of decomposition or DNRA. If it was the byproduct of decomposition, it could have added additional carbon and other trace nutrients as it liberated them from sources within the core. Or if it was DNRA, it could have increased this competition for nitrate and ultimately caused ammonia to significantly co-vary with denitrification. Finally, we have total nitrogen, which was significant at 2 of our field sites. This is a measure of the soluble forms of nitrogen, be it nitrate, nitrite, ammonia, or other organic forms that are found in an aqueous solution. And we found that this was significant due to high rates of mineralization or flux over the course of our incubations. Next, we evaluated 2 different forms of carbon. The first of which is dissolved organic carbon, or DOC, which was significant at 1 of our field sites. And then it's a measure of the labile forms of carbon. This acts as a readily available food source for the microbial community and was found to increase across all of our sites in all of our treatments, with the greatest increases being seen in our oxic cores, further supporting the idea that decomposition is ultimately taking place everywhere. Finally, we have ash-free dry mass, which is a measure of sediment organic material and was significant at 2 of our field sites. Ultimately, there was a significant amount of sediment in some of our cores, reaching as high as 10% of the sediment. But within our sandy sites, they were fairly organic lacking, making up only 1.5% of the observed sediment composition. And this is important as this organic material can be readily broken down, again, into more labile forms like DOC, but also release essential trace nutrients. Speaking of which, we evaluated phosphate, which was significant at all of our field sites. And while not directly related to the nitrogen cycle, it is, again, an essential trace nutrient with playing an important part in many microbial functions or components, including DNA, RNA, proteins, and many other cellular parts. Ultimately, we found that many of our field sites binded phosphate, and we found it was likely by two processes. First, abiotic binding of phosphate to charged sediments, such as clays, but also the luxury uptake of phosphorus by the microbial community when we saw binding in a field site that didn't have very much clay. There is an exception though. At our sand-rich sites, we saw phosphate liberation or the release of phosphate. Now, this was likely due to heterotrophic consumption of organic material, ultimately freeing bound phosphate that was found within the core. And finally, next I'd like to evaluate pH, which was significant at two of our field sites and increases as a byproduct of denitrification, largely due to proton consumption. It's important to note, however, that denitrification is optimized in these alkali pH ranges between 6 to 8, with our observed pH range being only slightly higher, between 7.5 to 9.5. But with regional variation, we think that these microbial communities may be able to acclimate and optimally denitrify under these conditions as well. But as denitrification can both cause an increase in pH, but also is optimized under these optimal conditions, it begs an important question. Are alkali conditions the cause or effect of denitrification? And this is ultimately something that we could not address in our incubations, but is still up for debate in much of the literature. Finally, I'd like to address temperature. And this is a fairly easy direct relationship, as higher temperatures equate to higher metabolic rates, as these microbial communities are ectothermic, with even minor changes having a detectable response in denitrification. We only saw about 1 degree in variation within our environmental chamber over the course of the incubation, and it still caused temperature to be significant at half of our field sites. It's also important to note that large swings in temperature are the primary aspect leading to variation in denitrification rates seasonally, which leads us to the larger portion of my incubations to reflect what these rates may be under different seasonal conditions. And ultimately, we found that temperature was the primary driver, as again, these microbial communities are ectothermic. And as you can tell here, the summer incubation facilitated our greatest rates of denitrification, followed by our spring incubation, fall, and then rates near zero during our winter incubation. While temperature did have this very strong effect, it's important to think about other factors that change seasonally, specifically wetting frequency, as the addition of water can increase the nutrients being transported into these wetlands, but also the nutrient load, as this changes seasonally, both through anthropogenic causes, farming, residential use, and the like, but also natural growing cycles, which may consume or release nitrogen based on what's occurring at that time of year. All the while, we can't disregard these context-dependent factors, things like the sediment composition, the amount of sand, ash-free dry mass, and many of the other factors I've addressed today, as they're constantly going to have an effect regardless of the season. Ultimately, though, what kind of insights can we glean from these trends? Well, I've shown you here that injection depth does affect denitrification rates, and this effect varies among different sediment types, with the greatest rates on average occurring at a silt-rich site. We also found, unsurprisingly, that anoxic conditions optimize denitrification, but within our incubations, we found that dissolved oxygen had the most variance related to it, followed closely by sediment traits, and then finally the nutrient composition, which we incubated all of our field sites with the same water. So this was a little surprising to see that it did have a significant effect. Ultimately, though, what kind of implications does this have on a larger scale? Because we need to be able to apply this. Well, we've seen significant sedimentation across the Dogtooth Bend Peninsula since 2011, specifically the deposition of 20.5 million cubic meters of primarily sand across the Dogtooth Bend. And that's depicted here as the various shades of green. Now, this is the equivalent of 15.5 Roman Colosseums worth of sediment being deposited primarily into existing wetlands. So for floodplain management, sediment deposition and type does matter. But we need to scale this in some way. If we were to hypothetically scale denitrification using our sand-rich sites based on the amount of sand that's being deposited across the Dogtooth Bend, what kind of rates would we get? Well, the Dogtooth Bend covers an area of about 90 square kilometers. If we assume only half of that area is inundated with flood water, what kind of rates could we observe? Well, assuming it's inundated with anoxic water, we could expect rates of denitrification to be as high as 29.1 tons per day of nitrogen to be produced. On the low end, this could be a mere 5 tons per day if these same sites are inundated with oxic surface floodwater. But we can't forget that denitrification does not always equate to nitrate loss. If we used or observed rates of nitrate loss, we could assume a removal of 8.1 tons per day of nitrate under those same anoxic conditions, whereas under oxic conditions, a mere 1.5 tons of nitrate removed. Thus emphasizing the necessity of these approaches if we're going to fully bound denitrification or observe nitrate loss in a natural setting. So for land management, what steps can be taken to enhance denitrification? Ultimately, this comes down to water control structures, as we need to inundate our wetlands with frequent, minor, slow-moving floods as to foster anoxic conditions, thus increasing nutrient consumption, but also increasing the efficiency of other control structures, as if we simply overwhelm them, they won't have an effect. And slowing these floods down will ultimately limit scouring, keeping much of our wetlands intact. Control structures that would likely have a significant effect within the Dogtooth Bend are things like tree plantings, check dams, and intercepting berms, as they would slow floodwaters, keep our wetlands wet, but also direct them into existing wetlands. Ultimately, though, there's room for growth, both experimentally as to explore methods as to replicate natural phenomena in the laboratory, thus allowing for higher resolution modeling to inform geospatial policy under climate change, but also to characterize regional variation of denitrification using modern methods and encapsulating the diverse hydrological conditions that we see in the natural environment. Ultimately, though, we need to bound these optimal conditions for microbial response relative to a few simple abiotic factors. Things like flow rate, oxygenation, and sediment type would be a great place to start, as the results would likely be widely applicable. And with that, I'd like to take a minute to thank the people and the organizations who have sponsored my research, specifically American Rivers and the Nature Conservancy, but also the undergraduates who have helped me analyze the plethora of samples collected over these last 2 years, Katie and Elise, and then my committee for quite literally helping me by getting their hands dirty, but also talking about the finer nuances of the science. And with that, does anybody have any questions?